series of forensic science lecture. Today we are going to discuss about the serology, the study of bodily fluids. So in this lecture we will be discussing about the blood spatter analysis, semen, urine and another biological fluids. These are the biological fluids which are found at the crime scene and they are very important in knowing about the victim and the criminal, as well as by knowing the various blood spatter analysis from the crime scene. We can get to know how the crime was committed, in what way the crime was committed, from where the person has used the weapon on the victim. It gives us a lot of information which will help us in knowing about the reconstruction of the crime scene. Blood is a very important evidence which is being found at the crime scene. The blood can be help us in knowing to identify or individualize a person. So if we have to individualize a person using a blood, we can go for the DNA analysis that can help us in knowing that this particular individual was present at the crime scene. After knowing that the particular individual, if we have to go for the reconstruction of the crime scene, we must look at the stain patterns of the blood which are present at the crime scene. These are also known as the blood spatter analysis. The distribution and appearance of blood stains and spatters can be used to interpret and reconstruct the events that must have occurred to produce the bleeding. So, if the weapon which is being used by the criminal to kill the victim will give us a different distribution pattern depending upon in what way, at what surface it was being used, at what force it was being used and which kind of weapon is being used. So, if any weapon like any arm or ammunition is being used by the criminal, that will have a different blood spatter analysis. If a person is using any sharp knife thing, then it will give a different stain pattern, and if the weapon which is being used by the person is like, for example, hammer, etc., that will give us a different blood stain pattern. So, the blood stain pattern depends on the harder and less porous the surface, the less spatter results. So, it generally depends upon the surface texture. We can also know about the direction of travel. The direction of travel of blood can be known by the stain's shape. The pointed end always faces its direction of travel. So, for example, if any person, any criminal has used a weapon, for example, a sharp knife like object, and if the blood is spattering over the particular area, so we can know that the crime has happened here, and from here the spatter has happened outside. So, the elongated shape will be outside of the blood. Another thing is the impact angle also tells us about the stain pattern. A drop of blood which is striking a surface at a right angle, it will give rise to a nearly circular stain, whereas if the angle decreases, from example from 90 to 60 to 50 to 30, the stain becomes more elongated in shape. So, they will have more like edge like thing, but if it is at 90 degree, the right angle, the stain will appear circular. So, origin of blood spatter, it occurs in the cases in a two-dimensional configuration which can be determined by establishing the lines through the long axis of several individual blood stains, and the intersection of the lines represents the prime point from which the blood is being emanated. If we are considering the reconstruction of the crime scene, we could say that the blood spatter is in the all directions. So, whenever we come back from those lines, then there is a point of convergence. We look at that point of convergence or the area of convergence, and we can tell that this is the particular point from the blood has been emanated, or this was the source, or this was the area from which the blood has gone, or the pattern has been formed, or the spattering has been done from this particular point. It depends upon till what distance covered by the blood drops, the velocity of the blood drops depends upon the starting from the resting position. Free falling blood drop continually accelerates and velocity increases. We say velocity is equal to gravitational acceleration multiplied by the time, v is equal to gt. Therefore, after 1 second the velocity would be 32 feet per second, and after 2 seconds velocity will be 64 feet per second. So, velocity is directly proportional to the gravitational acceleration as well as time. So, as the time increases, the velocity will increases. The first drops which are there will be nearer, and the second drops they will travel faster to the depending upon the first drops where they have been travelled. We also say a term in the case of blood spatter analysis, the terminal velocity. As blood drop falls, friction increases. When gravitational acceleration is there is equal to the friction and no acceleration, which maintains a constant velocity, which is known as the terminal velocity. The maximum terminal velocity which is being achieved is typically 50 microliter of blood is around 25.1 feet per second, which is achieved at a height of 14 to 18 feet. If we are talking about the volume of a passive drop of blood, it depends on the source from it formed and fell. So, the average volume is around 0.05 ml, or we can say it is 50 microliters. Another thing which is helping the blood to move is inertia. So, the net force is mass into acceleration. We know F is equal to ma. As the weight of blood drop increases, which corresponding increase in mass, the accumulation of weight of blood drop creates overall net force. So, with that inertia, with that spatter, the blood drops will go and form a spatter like pattern. So, there were studies which were performed by the Herbert McDonald. He has seen that the surface texture, whether it is harder or less porous surface, the spatter will be less. But if the surface texture is less harder, then the spatter will be more. And whenever the blood drop it hits a hard surface, it breaks up on impact casting of smaller droplets. So, we can say the first pattern, when we are at the crime scene, we are looking for the blood spatter analysis. We can easily say that this is the when the blood first hit the surface, this is the first pattern. For example, if a person is being killed and the blood is dropping from the top to the bottom. So, one pattern will be formed when the blood is being dropped to the surface. The second pattern will be formed when it impacts that surface and it makes another pattern on the surface of the floor. So, impact angle of blood is inverse of sine W by L, where W is width of stain and L is length of stain. So, as we discussed that if the angle at which the blood drop will fall, if it is at 90 degrees, then the blood will be more rounder in shape. But as soon as the angle will decrease, the blood drop will lose its round shape. The 70 degree it will appear round, but it will have the corners, more spooky corners. At 45 degrees, it starts to get elongated, 30 degree more elongated, and as the angle decreases more, the blood will lose its circular shape and it will have an elliptical shape at 11 degrees, 5 degrees or even at a lesser angle. So, if we are talking about and if we are going back to the point of origin from where the blood was being originated, we should know for the area of convergence on a two-dimensional plane. There are blood drops which are being spattered. So, we go back from those spatter, and where those lines meet, that particular point is known as the point of convergence, and the nearby area is known as the area of convergence. Convergence represents area from which the stains came out or emanated. So, the point of convergence on a 2D axis will be the x axis and the y axis, and we can see that two particular y axis where the blood drops are being spattered, and by knowing the distance which they have travelled, we can get to know what was the velocity of these particular blood stains. And we can get to know, if we come back from these spatters, as you can see in the image, we can go for the area of convergence, that this is the particular point from where the blood was being spattered. This is the area of convergence. At this place, the blood came out from the victim. Depending upon if we have the blood stains from the arteries, the pressure of bloods in the arteries is very high. So, the arterial spurts will give us the vertical blood stains on the wall. So, these are the blood stains which came out with a lot of pressure and which have a trailing ends also, and they are mostly found on the vertical surfaces. The cast of spatters can be shown that blood has been fallen down 90 degree from the top, and as they hit a hard surface, then they have form a secondary pattern, and it will show in which direction the blood has travelled, and we can know the angle. So, we can know at what speed, from what distance the blood has been fallen from the particular 90 degree angle, and from where this particular cast of spatter have been made of. So, these, when we are talking about the blood angles, the impact angles, by accurately measuring the length and width of a blood stains, the impact can be calculated using the sign formula, where the sign could be less than or equal to width over length. So, it's depending upon the width of the blood stain and the length of the blood stain, where we can get to know if the width is 1.5 centimeter, the length is 3 centimeter, then divided by width, we can get to know the sign is 0.5, the angle was 30 degree. So, impact angle from the blood stains can be found out using the width and the length of the blood stains. And when we are further doing certain chemical tests, we should know about the nature of the blood. It is referred to the complex mixture of cells, enzymes, proteins and various inorganic substances. The liquid portion is called plasma or serum, and it is composed of mainly water and accounts for 55% of the blood content. Whereas the solid portion is made up of RBCs, WBCs and platelets. There are certain antigens which are present in the blood, and they reside on the RBC surface, and they impart blood type characteristics to the cell. They stimulate the body to produce antibody against it. More than 15 blood antigen systems have been identified, which includes the ABO blood systems and RH systems. On what basis we say the blood group of a person, whether it is AB positive? When we are talking about the positive and negative, that comes from the RH factor, whether that particular RH factor antigen is present in your blood or not. These systems are used forensically to identify an individual. So, when we are talking about the ABA and RH systems, they help us in eliminating the suspects and help us in narrowing down the suspect list. We are going for the simple test like ABO and RH system based on the antigens, which help us in knowing and which help us in eliminating the suspects. Nature of blood also includes the antibodies. They destroy or inactive a specific antigen. They are found in the blood serum. For every antigen, there is a corresponding antibody. Antibodies only react with a specific antigen. The reaction is caused by the agglutination, or we can known as clumping. So, whenever a particular antigen is mixed with a particular antibody, there is a precipitin reaction. That precipitin reaction is known as the agglutination reaction. In such cases, when we are identifying whether the blood belongs to human or not, then we can go for the specific antigen-antibody reactions. Antigens are A, B, AB. If a person has A blood group, he will have antigen A and antibody B. A person with blood type B will have antigen B and antibody anti-A. A person having blood type AB will have both the antigens A and B, and it does not have any antibody, neither anti-A nor anti-B. Whereas a person having a blood group of O will have not either A antigen or nor B antigen, but it will be having both antibody A as well as antibody B. So, if we are talking about the principles of heredity, how this hereditary material being transferred from parents to the various generations with the help of microscopic units called genes. The genes is the basic unit of heredity and they are positioned on chromosomes. We have 46 chromosomes in human body which are in 23 pairs. This is the only exception that the chromosomes are not present in the reproductive cells. So, during the fertilization, a sperm and egg combine. So, each contributes chromosome to form the new cell zygote. Hence, whenever a new individual is born, it inherits the DNA or the chromosome from both the parents. The 23 chromosome pair will come from the father and 23 chromosome of one pair will come from the mother. So, the child which is being born has the DNA of both the parent as well as both the father as well as the mother. So, when we are going for the paternity testing or the kinship testing, then we can identify these particular material which is present on the chromosome to know about the parents of that particular person. So, genes that govern a given characteristics are positioned on the chromosome inherited from the mother as well as the father. Alternate forms of a gene that influence a given characteristic are aligned with one another on a chromosome pairs are called alleles. So, for example, if we are talking about the eye color, so the eye color of father is black and the eye color of mother is brown. So, the child might have the both eye colors, genes or alleles on his chromosome, but the color, the particular gene which is being dominant will show that color in the child. So, it might have alleles for both the particular trait, but the dominant trait will appear as a phenotype in the children. So, the inheritance rules goes by a gene pair of two similar genes is homozygous. There is no problem in determining a person's outward characteristics, but if a pair is heterozygous, then one gene is dominant, other is recessive. The person's outward characteristics depends on the dominant gene. If a person has the alleles for the same, for example, he has inherited the black eye color from both the parents, then the eye color of the child would be black only. But if he has inherited the black eye color from the father and the brown eye color from the mother, so depending upon the which particular gene is dominant, so if the brown is dominant, then the child will have a brown eye color, not a black eye color. So it comes in the cases when the gene which is being adopted is heterozygous in nature. So this is known as a Punnett square, where we know if the mother's blood group is AB and the father's blood group is O. So there are four possible combinations. These are the four possible combinations where the child could have either A blood group or B blood group. Because the antigen A comes from the mother and there is no antigen from the father, the child will have either A blood group, and if the B antigen comes from the mother and no antigen is coming from the father, the child will have B blood group. So, if the mother is AB and the father is O, then the child could be either A or B. They cannot produce a child having AB blood group or O blood group. So, that is how we can determine using the ABO blood grouping by using this Punnett square that what could be the blood group of the child using the blood group of the mother as well as father. So, genotype is the pair of allele of genes, whereas phenotypes is the outward physical expression of the gene, how that person will appear or what blood group will come into that particular person. Going next, if we are analyzing this Punnett square, 50% of the offspring will have chances of a genotype AO and 50% of the offspring will have chances of a genotype BO. We know that O is recessive, so 50% of offsprings have a type A blood group and 50% of the offsprings will have type B blood group. So, we can see from this example that no blood group gene can appear in a child unless it is present in at least one of the parents. As we have seen that in the previous slide, that a mother with a blood group AB and a father with a blood group O can have offsprings either with A blood group or with B blood group. So, this Punnett square helps us in analysis in the cases of paternity testing, through which we can tell whether this particular offspring belongs to parents just on the basis of the blood grouping. If the blood group matches, then we have to go for the further DNA testing or other testings. So, many cases of disputed paternity can just be resolved when suspected parents and offsprings are related according to their blood group systems. For example, the father's genotype is OO and the mother's genotype is AB and the offsprings could be either A or B. If the offspring father was blood type AB, would the suspect father be cleared? So, in that case, a child cannot have a AB blood group, which means that this is not the father of that particular offspring, because if the child is having AB blood group and we know that this is the mother of the child, then that means the father should not have either A blood group or B blood group, so that the child can have AB blood group. A father with the O blood group cannot produce a child with AB blood group when the mother is also AB blood group. Thus, these type of examples help us in knowing and solving the cases in the cases of Punnett square to know whether the offsprings belong to that particular father or to know about the paternity testing using this particular example. So, if the child was blood type A or type B, what we can say about the suspected father, that the father's genotype is O blood group and the mother's genotype is AB blood group. So, using the offspring blood group also, we can go back to know about the father's genotype if we know about the mother's genotype. In the paternity testing, routine paternity testing, it involves the characterizing many blood factors other than ABO. As we read that in the ABO blood group, for example, we can combine various other enzymes which is present in one of the father and which goes down to the offspring, and using these factors the probability of establishing paternity becomes better than 90%. But once DNA analysis is done, the odds become even better than 99%. Apart from that, another bodily fluid which is found at the crime scene is semen. A large number of cases involving sexual offences, making it necessary to examine the exhibits for the presence of seminal stains. The forensic examination of semen is a two-step process. Firstly, the semen must be located, and secondly, the test must be performed to prove its identity. It may even be tested for blood type. Because there are certain persons which release their antigens in other bodily fluids apart from blood also, and we can test blood group using semen, saliva, urine, etc. Semen is checked for blood type because many individuals are secretors. 80% of individuals are classified as secretors and they release their blood type antigens in that is in their saliva, semen, vaginal secretion and gastric juices, and we can identify for the AB antigens for the same. So, for semen location and identification, first it is readily visible on a fabric because it has a stiff crusty appearance, and at best this method is unreliable, although there could be the acid phosphatase color test which we can do to locate the semen and to identify that. Acid phosphatase is an enzyme which is secreted by the prostate gland, and it can be detected using an acidic solution and a fast blue B dye which shows a color change. And if the color change is positive, we can say that the semen is present. Another test which we can use for the semen analysis is Barberio's test. In the Barberio's test, we can confirmatory say about the presence of the semen, which is a crystal test like Takayama-Tickman test, which gives us the characteristic crystal shapes of the particular characteristic. Even if the fluid is fresh and being examined under the microscope, then we can see the presence of spermatozoa, the shape, head, tail, etc., which indicates the presence of the sperm inside that. But in certain cases, if the person or if the criminal is suffering from the disease called azoospermia or oligospermia, in such cases it gets difficult to detect the sperms in that particular microscopic examination. A positive result for acid phosphatase and a negative result for semen give us ambiguous results. So, in such cases, there are prostate specific antigen tests which can be used to detect seminal plasma. In such cases, if we have found a white color substance at the crime scene and we have to confirm whether it is semen or not, then we have to go for P30 prostate specific antigens. These antigens help us in confirming that the particular substance which is found from the crime scene is semen only. They can be found on the beddings or the object upon which the assault has taken place. The rape victim must be subjected to a medical examination, and we have to look for the pubic hairs, the vaginal swabs, blood, fingernail scraping, etc., and the clothing and urine. The collection should be done separately for the vaginal collections as well as the semen collections, and they should be sent as soon as possible to the lab. If we are talking about the timeline, living or motile sperm may survive up to 4 to 6 hours in the vagina of a living person, and a non-motile sperm may be found in a living female for up to 3 days and occasionally up to 6 days. So, when we are talking about we are taking sample from the female's vagina, then we look for that it should not be washed and look for the intact sperm samples. They are not normally found between 16 hours, but they have been found as late as 72 hours after the intercourse. So, we can know based upon these timelines whether the person was raped, and we can look for the further analysis to know which kind of body fluid has been found at the crime scene. The body fluids and the body tissue identification, they are important in the forensic science, and they help us in providing the key evidence in criminal investigations. They help us in establishing a link between the identification of the fluid and the DNA profile, which adds the further weight to this evidence. Analysis of all these body fluids which are found at the crime scene, for example blood, semen, saliva, they give us a robust evidence that this particular individual is involved in the crime, help us is knowing the victim as well as knowing the criminal, the number of criminals involved in the crime scene, and which edges us in the case and helps us in solving the crime. Thank you.